Welcome to another episode of Well, Don't Tell the Kids. I'm Janet Lee, your host, and today my guest is Dr. Heather Reed. Welcome you to the podcast, Heather.
Hi, thank you. Thank you for having me.
Heather is based in New England, where she is an associate professor at the University of Connecticut in Stores. Actually, I think you're in the D.C. metro area.
Yes.
For a visit.
I'm here for the Society for Neuroscience meeting, and I'll be here for the next week. Yes.
Okay. Are you presenting?
I am. It's actually quite an easy meeting for me because I'm only hosting 2 posters out of my lab.
You are an associate professor of psychological sciences and biomedical engineering, so two different, totally different departments.
Yeah. Those departments, many people in those departments rarely talk to each other, so I'm a bit of a rare bird.
Rolling back a little bit, I see that you earned your 2 bachelor degrees from Texas A&M in biochemistry and biophysics.
Yes. It's essentially a biophysics degree out of a biochemistry and biophysics department. But yes, That's exactly right. Very molecular, very low level.
Are you a Texas native or?
Yes, actually, my family's from Texas. I was born there, but I moved when I was 2 weeks old, and my parents were both PhDs, and so we traveled a lot all over the world. So I lived in Long Island, New York, and I lived in California. Grew up much of my life in California, in Monterey area, and but then Texas has always been home. And that's where my father was a professor there at Texas A&M, and it's also a family tradition to be part of that school.
So how did you get interested in the fields of at your undergraduate level?
The interesting thing is that because I was a biochemistry and biophysics major, I really didn't know what neuroscience was when I was an undergrad. As a matter of fact, I barely knew what a neuron was. I was studying molecules and electric currents, physics, lots of basic physics, even physics of ocean waves. I had classes in that, which I really liked, the mathematics and physics of ocean waves and fluid dynamics and stuff. And physiology and biochemistry is really very far removed in some respects from the kind of neuroscience I do today. So I guess I got sidetracked there. But basically, as an undergrad, I was in a very different world than I was after I left.
Well, walking forward from there, then you earned your doctorate in pharmacology, neuroscience in Chicago, Loyola University in Chicago, the Stritch School of Medicine. Can you just describe the research that you did there?
Yeah, actually. So I kind of ended up going there because I had discovered that I actually spent a little time before I went to Loyola thinking about what I wanted to do. So as an undergrad, I realized that I wasn't really excited about the molecular level of things. And so, I was, and I still am to this day. I'm excited about the molecular level of things if I can tie it into the behavior or larger properties of a system. Anyway, I discovered neuroscience. I had an aunt and an uncle. I had Jack Diamond, who was at McMaster University, and his wife, and my aunt Ann Forrester, who was also there. And they basically would talk, get family dinners and things about neuroscience, and I just started to get curious about it, and I took a summer course at Woods Hole Oceanographic Institute. Actually, it was at NBL, Marine Biological Laboratories, before I started grad school, and it just sealed it for me. I just said, this is what I want to do, is neuroscience. So then I went to work with somebody at Loyola who was doing, so it was a long path. I started at Northwestern. I got accepted into grad school at Northwestern.
Oh.
And
that's my alma mater.
Is it your alma mater? I love Northwestern to this day. Actually, I was accepted into the medical school, which was in Chicago proper.
Downtown, right?
And and I was working with John Disterhoff. I had started out as a technician, and I had gotten accepted into grad school, and I basically started working with him, and I realized that basically when I was offered a graduate fellowship at Loyola for 4 years, that I personally thought that was a better deal. And then I later learned that, well, they, at a lot of institutes, they offer you the first year, and then basically your advisor picks you up, and you go for 4 years that way, or you get on a training grant. And basically I didn't realize that, once you're in, you're in. And now I know this.
Right.
But back in that time, my dad was a plant geneticist, and so was my mom. And in their mind, you need to take the sure thing. And so I was offered a 4-year fellowship. I went there, and I actually loved it. I had a great experience. I really had amazing teachers, and my advisor was Nay Dunn, who was from Hong Kong, and he basically was a really amazing neurophysiologist and pharmacologist.
What was your topic there?
Yeah. So what I studied there was I actually wanted to develop this. I worked on developing this slice that would keep intact the dopamine neurons from where they begin in the brain. It's a certain kind of neuron that releases a neurotransmitter called dopamine, which is basically acting on sites in the brain that are very far removed from the cell bodies. And so the cell bodies are in one part of the brain, and then they have axons that leave and go to a very distant part of the brain. And so I was trying to make slices so that I could basically look at the physiology between the natural interactions between those pathways and their targets. And that was challenging because it turns out that the pathway in the real brain takes, in pretty much all mammals, it takes this large deviation. It goes from the middle part of the brain to the very lateral parts, and then it comes back middle again to where I was interested in looking at the frontal fields, the frontal parts of the cortex. So keeping it in the whole intact brain, like a dissected out whole brain, would have meant that I would have had to have a whole brain, which some people have done, but I was not doing. So anyway, I basically then worked on the neuropharmacology of the dopamine modulation of cortical synapses and activity. Yeah, well, it was, I guess the basic premise is to look at the synaptic physiology and pharmacology that underlies the dopamine regulation of frontal cortical function, which is involved in things like schizophrenia and executive functions that you might have, and certain types of memory that have to do with executive function.
Getting away from the purely molecular and moving into, you were describing it as more, a little more applied, or something where
exactly.
There was a context.
So for me, it was a big jump, right? But if we talk to my grandmother, this would still sound crazy, reduced. But basically, it was a big jump for me to move from the molecule, from single molecules and biochemistry, to now talk about, basically circuits in the brain.
Postdoctoral fellow at the University of California in San Francisco, and then a research scientist at University of California in Berkeley. So now you're somehow on the West Coast and maybe following down the same path?
Yeah. So I actually went from doing this kind of reduced synaptic physiology preparation as a PhD, and I did a short postdoc working on the neurophysiology that underlies visual motion perception, and so that was pretty big area. And then at UCSF, I looked at the now instead of visual perception, and I went in at sound perception. And basically the underlying theme there is I became interested and really excited about this idea that your brain is wired in some way and functioning in some way to process sensory information. And it's all organized. What I've discovered over the years in these different levels is that it's organized in terms of how you interact with your environment. It's very much organized that way.
Dr. Reed participated in a panel discussion at the University of Texas in San Antonio a bit back. I'm not sure the date. You were observing that an overarching question was, and I quote, how or why study sensory processing from a dynamical system standpoint as well as a neural coding standpoint. Can you break that down a little bit for us, like dynamical system standpoint versus neural coding, and why should we do this at all?
Well, the neural coding aspect, I think you might kind of say that the neural coding aspect of studying sensory processing is oftentimes looking at structure-function relationships in terms of, in the jargon of the field of neuroscience, we often talk about the Hubel and Wiesel approach, because it was inspired largely by this approach that they could go into the visual cortices and see the organized cortical columns that are actually have multiple levels of organization. You have a hypercolumn, which represents input from the left eye, and then also has a second half of that hypercolumn, which represents input from the right eye. But then if you look at it closely and you understand how information is organized in there, it actually turns out within the left eye ocular dominus column, you find that neurons are organized into clusters and mini columns, which are responding to specific little tiny points in visual space, which corresponds to activation of the retina through, a whole topographic organization. And so that's kind of a neural coding aspect of that. That's the structure part of the neural coding. And then when you get into the physiology and the computation that the brain does, you can talk about how basically in the visual system and in the auditory system, there are things that are computed. So in the visual system, for instance, when you see something out in the real world, basically if you see it with one eye, then of course it's monocular. And it's going to be going up through your brain pathways and activating all the synapses all the way up to this high level, perhaps at the primary visual cortex, for instance. And you're basically going to have one kind of input, but when you now open up your other eye, then you're going to activate pathways that ultimately converge on one another at some level of the system. And you can create and compute like a mathematical function. You're computing for the first time binocular information across that's picked up by the two eyes about the real world. And the same sort of thing happens in the ears. Right? So basically you have things out in the real world, some sound running past you, like a car going from the left to the right. And basically as it moves across your acoustic scene, your two ears are experiencing it in a different way, and your brain is actually computing that. And so the neural coding and theory part of what I've studied is looking at exactly how the neural pathways, particularly I've emphasized thalamocortical neural pathways, which is a certain part of your brain, and basically how they're anatomically organized and the different kinds of genes they might express in order to differentiate them from other pathways, and basically seeing how those are organized and seeing their synaptic physiology or their responses to actual sensory input, and seeing how the responses differ across different pathways, and that's the neural coding aspect. And we get into really nitty-gritty details, which I don't think is really interesting when you're talking to a general audience, right? But we talk about neural coding strategies, and so I've actually gotten into talking about how sound, like interest in the auditory system, it's kind of cool because we look at these maps of how sound is represented in the auditory system. So sounds are very cool in the sense that basically they're represented throughout much of the auditory neural pathways, very much like piano keys on a piano. So that your sensitivity to the low frequency keys on the left side of the piano is being transmitted through one pathway. And then as you move up to the high keys on the piano, that's being transmitted through another neural pathway. So they're topographically organized, very much like a piano keyboard throughout the system. And what I've shown in the neural coding and kind of structure function aspect is that the auditory system has a really interesting parallel and segregated processing where you get multiple representations of this piano keyboard in parallel. And if you look closer, what we find is that one region is actually it's a piano keyboard, but when you look at it closely, individual neurons are not so good at resolving the A note from the C note that's right next to it. They're basically poor at resolving individual notes right there by each other, but they can resolve the temporal information and sounds very well. And then we find another pathway that can resolve the notes. Each one of the notes on your piano basically is one semitone or 1/12 of an octave. And so basically, if you, in this other pathway, it can resolve that semitone. And that's an interesting property because now it can give you new information. And that's a new, so that's the sensory coding aspect.
Let me ask you something. Okay, so you're saying a temporal discrimination, or the able to resolve differences or different frequencies, I suppose. How do you see that in the lab? Is that a chemical thing where it's lighting up, or is asking as a layperson here? Is it by process of like a deduction or a process of elimination, or is it a direct observation of, oh, yeah, there it is. There's a stimulus and, just lit up.
Okay, well, so a little bit of all of that. Okay, so first of all, obviously when
curious.
I know. Well, you're an engineer, right? So
yeah, that's right.
And I've noticed that you have a little side interest in neuroscience, and so you bring the two together, and neuroengineering totally rocks. So first of all, sound is a physical property. It's a vibration. Right? It has to be transmitted through, some kind of medium, basically through matter of some sort, or water. You can talk about water. Basically, the lore in textbooks used to be you don't have a sound out in space. Well, they showed otherwise. In fact, when you have a star that explodes, basically you can get a wave of matter moving with the explosion, and sound will transmit with it. So you can get it out there in space, but you can't get it without matter. So the point is that that's the physical property, and you might be able to measure the frequency of a sound in the real world with an instrument that can measure those vibrations. But when we're looking to see if a neuron responds to a given frequency, or more reasonably a range of frequencies, then basically we're playing the sound to a subject. You can play this to humans, and basically you're then recording from their brain some sort of response that can be, like you said, direct or indirect. And so one direct method that we use is we can actually put tiny little very fine wires deep into the brain tissue and record electrical potentials from individual neurons using tetra technology. It's one approach. And then another thing that we're working on is less invasive approaches. So we can put a mesh of electrodes on the surface of the brain just beneath the skull, and then we've actually shown through that method and cross-validation with other methods that we get the same maps. So that's very cool. And then we can be even more indirect. So we can actually record metabolic changes that have to do with blood oxygenation. And what happens is, everybody knows when you look at your wrist, you see your veins, they look one color. And you know that if you were to accidentally cut your vein, or if you were to cut some surface blood vessel that's carrying unoxygenated blood, that the blood will go from being a blue-green color to being a red color. And that's because blood, the red blood cells have inside them hemoglobin, which has iron, and that creates a pigment or a color reflection and absorption spectrum. And so basically, as soon as it binds or unbinds oxygen, it changes the conformation of the protein just a little bit, and actually changes the way it reflects and absorbs light. So that's kind of cool. So you can take advantage of that, and we did basically using a technique called optical imaging, where we measure tiny little changes in reflectance, and what we see is that basically when an area of the brain is very active by a given sound, the neurons fire more. And it needs more oxygen. It needs more blood. And so you basically, and so that's one of the ways. So that's an indirect measure. So we've done it with all of these things. We can create these maps and map out that piano keyboard with many different kinds of approaches, which is very cool because it means that it's a very fundamental property and structure-function relationship.
Wow, that is amazing. So you work in the visual cortex area and auditory. Do you mix them ever together?
So I have mixed, like when I was studying visual motion, I was studying that in a higher level visual cortex called the parietal cortex, which, some people don't even really call it a visual cortex, but it's divided into different parts. That was something that a woman in science, Patricia Goldman-Rakish, kind of worked out, and later Liz Romansky. They worked out this idea that a lot of the different cortices are parcellated into kind of functional domains for one sensory processing than the other. But we recorded from this area that people thought of as visual parietal area, and but it turns out that it also has proprioceptive information about where your eyes are looking. So isn't that cool? So like basically you're in the parietal cortex, and it's activated when you move, when you look at, if you look in an aquarium and the fish are swimming off to one side, so it's going crazy, right? But then if you turn your eyes to look at the fish or not, it changes the map in the parietal cortex. It's very sensitive to that signal. In fact, it has a specific effect. You have what they call a multiplicative interaction between the map for spatial location of the image and the map for the angle of gaze of your eyes. And so that's a situation where you have two sensory pathways, the proprioceptive information from the movement of your eyes, telling your body and your brain that, I'm looking to the left and things are moving to the right. Well, that movement may be because I'm moving my eyes. And then the other situation is that when they're independent of one another, you can actually now reconstruct what they call, you can use this kind of information to reconstruct an allocentric representation of space from an egocentric map, that is where your eyes are moving. So just the idea that, so one thing that I have is becoming a common theme in what I'm seeing in these systems is that a lot of sensory maps, people think of sensory systems as being independent of motor systems because they do develop somewhat independently. You're certainly at the low levels of your nervous system, like your spinal cord. You have a ventral horn and a dorsal horn. And the ventral horn is where your motor neurons are that go out and synapse with your muscles, and they make your muscles contract. And the dorsal horn brings in all the sensory information, and it allows you to have a reflex. When you have a hot, you pick up a hot cup of tea, right? And so those are separate, but when you get to the high levels, it turns out that, your motor action of moving your eyes, it sends a sensory signal that you've just moved your eyes. And that sensory information converges with the sensory information about the visual input to your retina and gives you a very unique signal about what the object is relative to your egocentric or body-centered framework. So,
any guess why that is?
You actually touch upon something that's an interesting thing. So I was talking about the high levels, and that's where I started studying this. But people have actually, if you dig deep into the literature, it turns out that a lot of what we call as being unimodal or unisensory modal is actually already cross-modal at an early stage too. So so basically Eric Young, he's at Johns Hopkins. I think he's emeritus faculty now, but he's one of my one of the people that mentored and inspired me through just science conferences and going there and visiting his lab. But basically, he actually has a wonderful series of papers where they recorded from an early stage of the auditory pathway, and they showed that the neurons that everyone considered what they call the cardinal auditory neuron pathway, the limniscal pathway, which should only respond to sound and nothing else, that they actually responded to somatosensory input. And so it turned out that if you had if you move your head and turn and look to the left, basically the cells would shift how they're responding to a given sound. So they're actually, again, just like I recorded in the parietal cortex, the convergence of ocular motor and visual motion, these cells in the cochlear nucleus, which is brainstem nucleus, were responding and getting input from somatosensory neurons and auditory neurons. So, in fact, what you touched upon was you said, why should it be different? It turns out there's some of that early on that people do not really even know today. Like your average person doesn't know about Eric Young's paper. He's quite amazing, and people know a lot of his work. But that particular finding, they don't really recognize it.
It's consistent down through the levels, as you're saying, even showing
It's consistent to some levels. I mean, you have to be careful. Like with the retinal visual system, the output from the retina, basically it goes to the thalamus directly, and from the thalamus to the cortex. And basically, there are certain layers which people consider in the visual thalamus to be completely visual, and I don't know what's going on in that scientific field today, if anybody's ever found them to be cross-modal at that level for the visual thalamus. But in the auditory limniscal pathway, there's already some cross-modal input in the very first level. The more important theme is that your brain doesn't throw information away. And the things that fire together, wire together. And so if you're basically, if turning your head or turning your eyes is dramatically influencing the visual input for your eyes, or turning your head for your hearing, right? Then if those are coincident in time during development, when those pathways are wiring up, then basically they will wire in such a way that they converge at some point.
So that there's some efficiency going on, and also some robustness or redundancy, or that we take in cues in quickly as we need to, so that we don't get so we'll survive.
Right. Well, if you go to a honeybee, they actually converge things very early on. So there is a bit of that, the concept that you alluded to, this idea that things in the mammalian system actually tend to be highly segregated, and then you get some convergence of a sort happening only higher up. But in a bee, for instance, I've actually recorded intracellularly from a cell in the bee that does visual motion, but that same cell responded to, again, movement of the little bee head. And so basically, it was a visual motor neuron. It was sensitive to the movement as well as the vision. I don't know if you would really call it motor, but it was certainly proprioceptive in a sense, right? And so the bee is going to be quite different in terms of where that convergence might happen and how it happens.
Let me just shift gears a little bit here.
Okay.
You also have a leadership role in something called the Brain Initiative. Could you maybe describe that, its mission?
I'm honored that you would say I have a leadership role. I think that a lot of people there's been amazing efforts in order to make this whole initiative happen, and my role has just simply been somebody who goes in there and helps review proposals and helps determine, and also to chair the review panels in order, which is interesting, and that's another topic, but we can talk about. But first, let me just say what the Brain Initiative is. So the acronym refers to the brain research through advancing innovative neurotechnologies, stands for.
BRAIN.
So brain research through advancing innovative neurotechnologies. So what does all that mouthful mean? Well, I mean, the idea here is to inspire a really new approach to neuroscience, right? And so the idea is to bring together a new approach and to create new methodologies for basic research, and then to use this approach to understand the brain better, and hopefully to build better approaches for diagnosis and treatment of neurological and psychiatric disorders like Alzheimer's, autism, epilepsy, depression. That's kind of the big overarching goal, is to get at the basic science so we can have a framework for which to better all of our diagnosis and treatment.
Where is this being funded out of?
So that's kind of a nasty question, because it's funded out of a lot of institutes. And I'm going to have to say all those names. But I can tell you the National Institute of Health, for instance, and the National Science Foundation, the Food and Drug Administration plays a role in it, because the goal is to come up with new methodologies, and some of these methodologies are being applied to humans. And so if we're going to, like, let's say in the future, we're going to come up with a device, which we, which in my lab we've researched it a bit, a minimally invasive device, but it still goes into the skull. That situation, it's minimally invasive, but for a human, you really must have it go through the FDA, the Food and Drug Administration, in order for it to be considered low enough risk for that to happen and high enough benefit. Yeah. It also includes DARPA and IARPA, by the way, and some private partners. I just want to throw those in. I don't want to leave anybody out.
You're the gatekeeper, or one of the gatekeepers, I suppose. Anything striking that...
So I think what we're witnessing, and I think what the BRAIN Initiative kind of really rode the wave, so to speak, of, is this change in science, right? And so it used to be when I grew up and when my parents were PhDs and when they were growing up, science was a single man in the lab with a few people working for him. And now it's becoming many people working together to solve problems. And so I think that that the cool thing is some individual grants and proposals that I've seen come through really bring together people from all walks of life and expertise. The idea is that it gives you this fertile environment for creative and innovative development of technology for neurotechnology, right? And so, for instance, you might have a proposal that's going to bring together neurosurgeons, and we did see this. So it's the neurosurgeons, epileptologists, and linguists, or computational neuroscientists, or maybe people who do development of a certain behavior, and bring all those people together, and then they have to solve a common problem, a question about a brain structure-function relationship or something like that, right? And so in humans, so that's one of the goals for the BRAIN Initiative too, is to have impact on human health, right? And so so bringing those people together is crazy. And so what it means is the review panel consists of also an equally diverse group of people. So I had people in there who were my icons, people who I was just like, oh my gosh, this person's sitting at this table across from me. And I've always just been in such awe of their work, and they're sitting right there, and I've got to make sure that he stays on time with what he's saying, with his synopsis, and that everyone is synthesizing the most cohesive review, that we get the best thing possible out of this session, right? And so it's kind of overwhelming. And then also it's beautiful because you have neurologists informing neurophysiologists who really, you'd think it sounds like they might understand each other's field, but sometimes they don't. And so they're having to explain to each other the shortcomings of one approach versus the other. And so that's a very interesting thing. So not only is it diverse in terms of the proposals that bring together people across disciplines, but at the table, when we're having to make a call, a judgment call, you have an equally diverse and well-informed group of people to cover things like, risk. We want to make sure that there's low risk to human patients, right? And then you also want to make sure that it's of high potential benefit to society and to humans. So,
how long have you been involved with this initiative?
So this particular initiative, I've worked with them for only 2 years, but I've worked with other review panels for that kind of lead into this because it is across different agencies. Yeah, so I've worked for NIH and NSF panels before.
Just going to ask if you've seen a change over a period of time, long or short, in the diversity. You're speaking of intellectual diversity, which is which is necessary to respond and to decide what goes forward and whatnot. But have you seen any diversity in terms of women in science?
I tend to be a positive thinker, and I am hopeful that things are getting better. But I think that it was interesting because the National Science Foundation, I really think that they have made great strides in trying to make it a fair playing ground for including people across all kinds of diversity fields, but certainly for gender. And so I've sat in there and had program officers and everybody really lecture us and say, set down the law that, this is that you don't bring your biases into the room. And that's actually very, very welcoming. Now, whether or not there's still imbalance in terms of gender in those study sections, unfortunately, there still often is. I mean, I've been on NSF panels which were very equally distributed between males and females, and I've been on others where they weren't for whatever reason, and sometimes like certain fields tend to be just dominated more by males, like certain like neuroengineering and neurocomputational fields, which some of my grants go to. Some of them tend to be dominated by males. Yeah. I think it is. I think it's cautiously encouraging, and I think the important change point in a lot of these kind of bias issues in science, I also have been involved with Bias Watch Neuro, which basically a lot of men and women are involved in. And the concept is to come in, if we don't get both men and women involved in trying to change this bias, then we won't make any progress, because, it's got to be at the basic. It has to be a basic goal for all of us in order for it to move forward. And I think it is. I think people are starting to really be supportive.
I think you may have an involvement also with some outreach, working with or encouraging the youth. So is there anything that you would say for that portion of the audience?
Yeah, I was the director for the Women in Math, Science, and Engineering. They always have these funny acronyms for these women in STEM groups. Like I was WIMSY, and I think there's one at Cold Spring Harbor Labs. I think it might be WISE.
Wisdom and WISE, yeah.
For incoming people, like newbies, undergrads, yes.
We're just people contemplating, coming down this path.
Okay, I learned a big lesson. So when I first started teaching undergrads, not working with undergrads in this area, I was actually working with women who were first generation in STEM fields, so they hadn't been in physics or math or engineering before, or even neuroscience, and they were they were the first one in their family maybe to go to college, and so I wanted to just tell them everything. Like, I wanted to tell them, like, you better look out. Like, I remember one of my advisors told me, oh, yeah, things might seem really equivalent down at this level, but by the time it moves up and resources get fewer and fewer, things are going to get tough. And I remember kind of seeing a little bit of that. But, I wanted to tell them, like, oh, it's tough. And then, if you have a family, that's even, that's difficult. There's all these pulls on you. So you really need to be focused and get going. And I realized they don't want to hear all that information at once. Nobody does, right? And so I learned that you have to just inspire genuine curiosity and excitement and help them find what is going to be exciting for them. Now, for a first, semester, first year STEM, first generation STEM person, that it can be overwhelming, right? They're not, it's hard for them to keep up with all the schedule of exam taking and the new concepts that they're learning, right? Sure, and so it can be overwhelming. And so some things that you have to guide people on is just really practical things like schedules and keeping track. This is your goal, then let's work back from that goal, right? So if your goal is to make an A in this class, and in the final class, and how many exams do you have? Well, let's go back to that exam. So, okay, what do you have? You have 5 weeks before the midterm. Okay, so if that's the case, then what are you going to do the first week? Right? And it sounds so mundane and horrible, but a lot of these particular people really needed that. And then the beautiful thing is that when I, tried to mix up the kind of get them excited by talking about basic wonder of science or neuro engineering or physics, when I tried to just get them excited about that stuff by bringing in speakers in specialty fields and also speaking myself, I found there was, different people rose up and learned from each one of those things. Some people really thrive from, me telling them, hey, you need to organize your schedule. And other people thrive from being inspired by the science.
It's not all glamorous.
It's not.
And then also just getting through the mechanics of proposals and grants, those are valuable lessons just to learn early.
Oh, yeah. Yeah, we do. We do teach them that too, to how to, tell them about what are, they're basically constructing a basic proposal. Even at the undergraduate level, we've worked with them on that.
But thank you so much again for your time, and I hope you have a productive, constructive time in the nation's capital with your conference. Well, I think we'll wrap up here. So thank you, Dr. Reed.
Wonderful. I really enjoyed listening to your podcast, and it was a lot of fun to meet you finally and talk with you.
Thank you all for listening. My guest today has been once again Dr. Heather Reed. I'm Janet Lee. So please stay tuned for more episodes of Well, Don't Tell the Kids.